Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. Michael Skinnider. Listeners, Mike is assistant professor in the Ludwig Princeton Branch and the Lewis-Sigler Institute for Integrative Genomics at Princeton University. And today, we're looking forward to discussing his career and his research, including an innovative research project focused on the development of a chemical artificial intelligence platform for discovering designer doping agents. So, Mike, we're excited to have you here. Welcome to the show. And how are you today?
I'm doing great. Thanks for having me.
Well, we're looking forward to getting into some of the details of your work, but let's get to know you first. So can you tell us a little bit more about yourself and how you got interested in research to begin with?
Sure. So I grew up on the west coast of Canada in Victoria, British Columbia, and was very interested in both science, but also the humanities growing up. And so I went to McMaster University in Ontario, Canada for my undergraduate degree. And I was in a very interdisciplinary arts and science program there and kind of leaning towards doing something humanities adjacent until at the end of my second year, I got a position in a lab in the biochemistry department and realized very quickly that this was what I wanted to do with my life. So that all just kind of clicked. And even now I'm working on, I guess, similar problems to what I first started my career working on as an undergraduate student. So I was very lucky to have that opportunity.
I love that. Well, it sounds like working in this lab as an undergraduate was a pivotal career moment for you. What then were some of the steps that you took to get to your current position there at Princeton?
I ended up working in that lab for a little under 3 years. We were working on basically discovering new antibiotics produced by bacteria. That work involved developing computational tools to take bacterial genome sequences, predict the small molecules that those bacteria make, and then find those small molecules in metabolomics data from the same bacteria. After I graduated, I moved back to the west coast of Canada to Vancouver to do my MD and my PhD at UBC. My thesis work was, again, largely on computational mass spectrometry, mostly for proteomics, but with a little bit of small molecule work sprinkled in. I also spent some time in Switzerland as a visiting PhD student working on a very different kind of flavor of computational biology, working on single-cell genomics of spinal cord injury. And after finishing my PhD and going back to med school to finish my MD, I ended up starting my lab here at Princeton in the fall of 2023. And we've been working on all sorts of problems in the general space of discovering small molecules with spec since then.
Well, that's awesome, Mike. It sounds like you've had some really cool career experiences already. Can you tell us a little bit more then about your current role and just the scope of the research that you do in your lab right now?
So I'm an assistant professor in the Genomics Institute at Princeton, the Lewis-Sigler Institute, and then in the Cancer Research Institute, the Ludwig Princeton Branch. The lab as a whole is sort of broadly interested in developing computational and sometimes also experimental approaches to identify small molecules in the world around us and in our own bodies, and increasingly also in sort of understanding the functional roles or biology of those small molecules. One of the major motivating problems for the lab as a whole is to discover endogenous small molecules or exogenous small molecules that are associated with cancer, either the diagnosis, progression, or treatment of several different forms of cancer. But the technologies that we're developing are very fundamental, and mass spectrometry in particular is a technology that has so many different applications that these technologies can work for many different classes of small molecules. And so that's where the connection to anti-doping comes in. The same tools that we're developing to try to discover, for example, oncometabolites, you can just as easily take those tools and apply them to look for doping agents.
Very interesting. And I think you mentioned that there's this connection, but it's not obvious. Your work has been focused on genomics and cancer and these more biological problems. But how did you discover that there was this connection with anti-doping, that your work could be relevant there?
Yeah, it grew very organically out of some past work, which has really set up what we're aiming to now do in the anti-doping space that was addressing a fairly related problem of novel psychoactive substances, also called designer drugs. The similarity there is that, like new doping agents, designer drugs are often made by making a small modification to the chemical structure of an existing drug. And those modifications are typically made with the express aim of avoiding drug control legislation or the detection of those drugs with, for instance, existing screening techniques. So we had developed this platform to discover new designer drugs. And one of the toxicologists that we were working with on that study had actually done his PhD in an anti-doping lab, and he still kind of had an interest in doping. And so this was actually years ago, but he put it in our heads that we could use very similar approaches in the anti-doping field. That's sort of the genesis.
That makes sense. And then how did you first hear about the Partnership for Clean Competition and decide to apply for research funding?
It was actually from that same toxicologist. It was from Dan Passon, who had done his PhD in equine anti-doping lab in Australia, run by Adam Cawley. So he not only put this idea in our heads, but he also specifically said it might be worth applying for funding from the PCC.
Very cool. And you were recently approved for a PCC grant working on this novel chemical AI platform focused on discovering new doping agents. So I'd love to talk about this project in some detail. Can you maybe first share the potential value of being able to identify and predict some of these designer drugs that could eventually be showing up in sport?
So, I mean, coming back to the NPS work where these designer drugs being used recreationally rather than in the context of athletic competition, we had found that we could use this generative AI approach to predict the structures that clandestine chemists are likely going to synthesize in the future and then introduce onto the illicit market. That, I think, is the same value that we're trying to bring by deploying similar approaches, but training them instead on the structures of performance-enhancing drugs. I think historically, it's often been the case that a new performance-enhancing drug is some sort of minor structural modification of an existing agent that allows this derivative to evade analytical methods for the detection of known agents. So I think that if we could predict which hypothetical future doping agents might be used either now and we just don't know about them or in the near future, we could conceivably search for those agents in samples from athletes and we could detect them much sooner. That's the hope anyway.
Absolutely. I think staying ahead of what's happening down the line in the anti-doping field is critical because it takes time to develop the detection methods and the downstream things that you need to be able to have a robust anti-doping program to be able to detect and deter use of these new things that are coming on the market.
Yeah, the deterrence piece in particular is something that I had a chance to attend a recent PCC conference, and that was something that several people thought would be valuable in and of itself. Just the knowledge that there are tools that are specifically developed to search for agents that may not be on banned lists explicitly. So hopefully that, again, has value in and of itself.
Certainly. Well, let's dive into some of the nuts and bolts of the project next, Mike. Can you share what were the aims of this specific project that you submitted to the PCC?
So there's two main aims. The first is that we want to develop a generative model to predict the chemical structures of PEDs that are likely to emerge on the market in the future or conceivably are already in use and just haven't been discovered yet. And to do that, we're going to be using a framework based on language models. So the same kinds of neural networks that have been very successful at learning the syntax and semantics of human language. But instead of applying those neural networks to model, for instance, the words in a sentence, we're going to use them to model textual representations of the chemical structures of known performance-enhancing drugs. So that's aim one. And then the second aim is to develop another AI model, this one to predict various different analytical properties of these hypothetical PEDs. In particular, we'd like to be able to predict their MSMS spectra. And the utility of this second model would be that if we have a series of structures of PEDs that we hypothesize might exist or might exist in the future, and we have their predicted tandem mass spectra, we could actually go searching for those in samples from athletes or ceased products, et cetera, and try to establish whether they're physically present in these samples.
Very interesting. And it sounds like these drugs that you're predicting are based on small structural changes to things that already exist. What makes something more likely versus less likely when you're doing these predictions?
I think that both in the field of doping and in the field of toxicology, these derivatives have a certain logic to them. They're being made by medicinal chemists, and those medicinal chemists have a toolbox of chemical transformations that they like to apply. And our past work has shown that it's pretty straightforward to learn that toolbox based on the structures of drugs that have been introduced into the market already. So if you have a series of steroid derivatives, they all have the same scaffold. They have many different small structural changes at different sites on that scaffold. That tells you a great deal about which sites are important to modify or less important, and how exactly one might go about making those chemical modifications.
That makes sense. So let's talk about the team here. Who will be contributing to this project?
The plan is to hire a postdoctoral fellow to lead this work.
And the two of you will be working closely together on it, it sounds like.
Yeah, exactly. And we're fortunately in a position where because we have this large body of prior work, many of the methods are fairly well worked out. And not only practically, but we've done some work on the theory behind these methods. So I think we're in a good position.
Definitely. And I'd love to talk about the methods in a little bit more detail, as well as sort of sounds daunting to be able to build this whole complex AI platform that's able to cover such a wide range of potential designer drugs. So how did you go about doing this?
It's been a very, very long-standing interest of mine. Like I mentioned, 10 plus years ago in my undergraduate research, we were kind of using rule-based methods essentially to try to predict what structures are most likely. And we were certainly not the only people trying to make those rule-based approaches work. But really, in practice, it seems like language models have allowed some major breakthroughs in generation of small molecules. So yeah, the basic idea, like I mentioned, is to represent chemical structures as short strings of text. We use a format called SMILES that's actually been around since the 1980s.
Oh, cool.
And then we train what are called autoregressive models on these textual representations of chemical structures. So the idea is the model learns to predict what the next essentially character in one of these SMILE strings is most likely to be. Typically, each character represents an atom or bond. So it's a bit like asking the model to predict, okay, maybe you've seen half of the structure. Predict what the next atom or bond that needs to be added to this structure is going to be. And then once the model is trained on the training set of known here performance-enhancing drugs, we use a similar autoregressive approach to sample new SMILE strings and therefore new structures. So we kind of go character by character, asking the model to sample, again, you can think of it as sample a new atom or bond based on the sequence of atoms or bonds that it's already generated. And there's a couple sort of key ingredients to this to talk about. So one is that in these settings, the amount of training data is typically pretty small. So you think of large language models being trained on all the text on the internet, but here we're limited to 1 to 2,000 performance enhancing drugs. So one of the key ways that we kind of train a pretty sophisticated model on this relatively small data set is we take advantage of the fact that a given structure can be written out with many different short strings of text. So you can generate typically hundreds to thousands of different SMILE strings that all represent the exact same chemical structure. So we use that as a form of data augmentation. We generate many different SMILE strings that all represent the same structure. And in this way, we can functionally turn a data set of 1,000 structures into a data set of 100,000 or 1,000,000 strings of text. And then the other thing that's important to note is that once this model is trained, we've found repeatedly that when we're using it to generate new structures, models such as this don't generate structures with equal frequencies, but rather they disproportionately generate structures that the model views as particularly likely extensions of the training set. So what this means in effect is that the model assigns to each generated structure a kind of quantitative score that you can think of as its prioritization of this particular structure. So basically, this allows one to separate the generated structures that the model views as very likely, which are disproportionately likely to emerge in the future, compared to the structures that the model views as perhaps less plausible guesses.
Well, that's really helpful. And that actually answers part of my next question, which was, if you get such a large bank of data coming out of this, how do you prioritize it? How do you make decisions about what is worth pursuing? So can you talk a little bit about the anticipated outcomes and what you're going to take away from this project?
To that point, I'll also mention that one of the other big ways that you can make that decision about what's worth pursuing is using the second aim, which is the predicted tandem mass spectra. And the idea there would be if you have some signal in a sample or in a data set that you think is anomalous or you have some suspicion of, you could take that signal and use both the predicted mass spectra that we're aiming to generate as well as the prioritization from the chemical language model and use that to come up with a short list of candidate structures that are likely to explain that signal. In the toxicology space, we showed that that approach was really quite accurate, I should say. So we tested this combination of the predicted MSMS and the language model prioritization on a set of about 80 designer drugs that were introduced or discovered after we had trained these models. And we found that we could get the exact structure of one of these new drugs correct about 50% of the time. And we ranked the correct structure in our top 10 predictions about 90% of the time. So the implication there would be that if you're interested in a particular signal, 9 times out of 10, that correct structure would be somewhere in a short list of just 10 candidates. So this brings me to the other part of your question, which is what are we hoping to provide? And the goal is really to give the community access to this technology such that someone working in an anti-doping lab could take that suspicious signal and submit it to a web application and get back the short list of likely structures that I just mentioned. I guess I'll say, just since I was talking about the performance of this approach in the recreational drug setting, I wouldn't want to guarantee that we're going to be able to achieve the same level of performance in anti-doping, but it's a strong basis to think that this can be a useful resource. And the opposite is also true. It may be that we're even more successful.
Very cool. And I guess in terms of thinking about the practical use of it, it sounds like people could kind of upload their data eventually to this platform. How quickly would they get a response? Would this be like immediate?
Pretty much. We've done something similar in the mammalian endogenous metabolite setting, and we have a working web server up now for that. And it's not quite immediate, but it's well under 5 minutes.
Oh, wow.
So it would be pretty rapid.
Effectively immediate, we'll call it.
Yeah.
And then in terms of the timeline for the overall project for this anti-doping piece of it, can you talk a little bit more about what that looks like?
So I guess in the first year of the project, we're hoping to focus mostly on the language model. And then in the second year, the MSMS prediction, and then getting this web app up and running. That's the goal anyway.
The order of operations. I like it.
Exactly.
And we touched on a little bit how this work could impact the field of anti-doping. Maybe could you think about more broadly how people will use it, how you'll implement it, kind of how you'll get this into routine everyday operations?
I think to answer that, it'll be important for us to try to work as much as possible with anti-doping labs. What I'm envisioning is that once we have sort of a prototype of this system, we would do our best to connect with some of the anti-doping labs that would be interested in this sort of technology. And there are some people from the PCC who have offered to help make those connections. And we've actually talked to a couple labs already. But I think the goal would be to put this prototype out there and then to talk to anti-doping scientists about, is this meeting your needs, or is there other data that we should be incorporating? Are there other types of predictions we should try to make? Are there changes to the user experience that would make it easier to work with? But I think that probably the people who are actually doing this work are the ones who should be guiding that step of the process, ideally.
Definitely. And I think oftentimes you can't fit everything into one research grant or one research project.
Yeah, exactly.
So let's talk a little bit about the future directions. Mike, where would you take this then next in terms of moving this line of research further?
Again, I think I'm very keen to hear from anti-doping scientists about what they think would be most useful. For me, two things that jump out as being particularly important would be, one, the ability to predict metabolism of these structures. So we have a few drug metabolites in our training set. When those metabolites are named on banned lists explicitly, or when we've been able to go through the literature and establish that, okay, here are long-term metabolites that people test for that are not themselves in banned lists, but which are accepted as evidence of ingestion of one of these agents. I think that probably leaves a lot of metabolic transformations of these drugs on the table. So maybe an important next step would be to try to make that data set a bit more systematic, i.e. curate what's known about the metabolism of doping agents and perhaps prescription drugs more broadly, and then try to automate some of those predictions so that we can search not just for drugs and a subset of their metabolites, but more holistically for many possible metabolites of these drugs. And then a second avenue that's on my mind is being able to predict other analytical properties beyond tandem mass spec data. So tandem mass spec data is nice because although mass spectra produced in different labs will look dissimilar to one another to a certain extent, they also have much in common. But other analytical properties, for example, retention time, the value measured in one lab could have almost no relationship to the value measured in another lab, depending on their specific chromatographic setup. So I think if there's interest from the community and there's interest in sharing some information about the chromatography that people are using in the field, then we could think about predicting retention times for these hypothetical compounds and then integrating that retention time data with the other suite of properties that I've already talked about.
Oh, very interesting. And I think developing an entirely new kind of platform comes with its own challenges, and there's always limitations. So let's talk about those as well. What are some of the challenges that you've faced, at least thus far? And what are some of the limitations that you anticipate with developing and using this kind of AI platform?
Well, I think the biggest limitation is that this approach fundamentally is designed to interpolate rather than extrapolate. What I mean by that is this method learns from the structures of known doping agents and predicts likely transformations or derivatives of those agents. It's not going to dream up a completely new scaffold that's never been established as a doping agent and spontaneously think that into existence, which is an important limitation. Because if a new drug is introduced with a, for example, totally new pharmacological mechanism of action, we're not going to be able to catch that using this sort of platform. On the challenges side of things, I think I mentioned really the biggest one, which is just that the training data set is pretty small, but we have a lot of prior work showing that we can get around that. I guess a challenge on the mass spectrum prediction side is that this is a very long-standing open problem. So we're using some of the most cutting-edge ideas that have been applied to the challenge of predicting tandem mass spectra from chemical structures, but those predictions are by no means going to be perfect. So appropriate caveat to all of what I've said would be that ultimately these tools are designed to help raise and refine suspicion by experienced anti-doping scientists. And ultimately, these are predictions that require experimental follow-up and confirmation. And certainly I would not want to sound like I'm advocating that they be used to actually make decisions about athletic competition. These are really hypothesis generating tools.
I think that's really important. And I think just in general, I think most of us who have used AI in some capacity have seen the things it generates that aren't quite right, whether it's 6 fingers in a picture or descriptions of things that are not actually true. So can you talk a little bit about how these checks and balances are built in there, or how you're going to be able to convey that this is something that you do need to double check?
On the quantitative side, we are planning to provide a series of different, effectively, confidence scores to the user so that the user can appreciate, okay, how likely is this particular structure in the context of the training set of known PEDs? How well does this spectrum match to the experimental spectrum? What's the overall level of confidence that this structure matches the suite of analytical data for this particular signal? And then qualitatively, I think we just have to repeat this point that I just mentioned, which is that ultimately this is a hypothesis generating tool. And my dream in that respect would be that we can actually work with a doping lab, synthesize or order a chemical standard for a predicted structure and confirm or discredit it. And I think that would be a really nice case study to exemplify the idea that this is a useful tool. It can generate ideas, and many of those ideas are likely to be good, again, based on our past work in related fields, but it still requires confirmation using rigorous analytical protocols.
Absolutely. And I think just thinking about the big category, if you will, of performance enhancing drug, there are a lot of different types of drugs represented there. So Mike, does this kind of a platform work better with certain classes of drugs or certain starting scaffolds, if you will?
The big point that I should make there is that we're focusing entirely on small molecule drugs. And that obviously leaves a number of important and emerging classes of doping agents out of the picture altogether. Beyond that, in terms of specific classes, I think these approaches work well when there's lots of training data and they can learn the relationships between different structures in the training set. I would guess that our model is going to perform pretty well for things like anabolic steroids, where there are many examples of banned substances. It'll perform less well for compounds that don't have any similar compound in the training set. So perhaps certain kind of SARMs where there isn't a broad structure activity relationship literature, or we're just not aware of that literature in the sense that those compounds aren't on banned lists and therefore they're not in our training set. Cases like that, where there just isn't a lot of data, those would be the cases that I think we may produce less good predictions unless the kinds of transformations that are applied to those compounds end up being very similar to the kinds of transformations that are applied to chemically unrelated structures in our data set.
Well, that makes sense. And I think we've covered some of the important caveats and limitations, but I'd love to talk about the strengths as well. I think this is a particularly innovative approach that has a lot of potential. So can you talk about what you see as the strengths and maybe what makes this particularly novel?
For me, the defining strength is that the challenge of having an unidentified peak in a mass spectrometry data set, and you have some auxiliary information that makes you think this is an important signal and you would really like to know its structure. The scale of this problem, it's hard to overstate. If you naively think about every possible structure that could explain a given accurate mass measurement, you're talking about an astronomically large number of structures. And so I think the ability to narrow down that chemical space and to generate ideas that are good or even correct has a ton of value. We've seen that in our prior work. We just published a paper, again, doing something very similar for the discovery of endogenous mammalian metabolites. And we have another large project that we're trying to get ready to submit. And in these projects, we've been able to use these approaches to discover just a huge number of previously unknown small molecules that are made by humans or mice. And this is something that historically has been really challenging to do. So I think that I hope anyway, that giving anti-doping scientists a tool to get the ball rolling on this process of structural elucidation of totally unknown compounds, my sense is that that would have a lot of value. The other thing, I guess, is that both in the field of recreational drugs and again in the field of metabolites, by assigning each hypothetical compound this quantitative score, we've shown that these models can actually forecast the next wave of structures that are going to emerge on the illicit market or be discovered. So I think that that could have a certain amount of value in and of itself. This might be a set of predictions that agencies like PCC would be interested in. Perhaps the existence of these predictions spurs action in some respect.
Certainly. And you mentioned, Mike, specifically, you're focusing on small molecule drugs at the moment. Could a similar approach be applied to larger molecule compounds, or is this something where it would just become too complex?
It definitely could. I think just the sheer diversity of different classes of agents that one might like to cover and the relatively limited training data for those would be pretty important obstacles, but it's something we can definitely chew on over the next couple of years.
I like it. Well, it sounds like you've made a lot of exciting progress on this project and in this line of research in particular, but I know collaboration is critical in science. So I'd love to talk about how you work with other stakeholders, whether it's other researchers, you mentioned tapping into these anti-doping laboratories, as well as funding organizations like the PCC. How do you work together in this work?
We develop tools because we want to see them used and really make a difference in the real world in different fields. So for me personally, one of the single most satisfying things is to actually help put these tools into practice, work with experimentalists to validate a subset of these predictions. That's something that I'm really looking forward to and very much hoping that we can do once we've gotten the computational methods off the ground a little bit for this project. We're relative newcomers to the field of anti-doping, and I think it's important to get input from the sort of target audience for these tools and hear from people about, is this tool making reasonable predictions? Are there obvious avenues to improve it? And is it producing output in a format that's digestible and accessible and useful?
Fantastic. And you mentioned earlier in our discussion that you were able to, last year, attend the PCC's conference in New York City. So let's talk about that experience. What was it like really diving into the deep end of the anti-doping community, getting to meet with people and getting to share your work?
It was a lot of fun. It was really great to get a bit more perspective on how people in the field think about these problems, just learn sort of the language that's used, what the most pressing issues are perceived to be. I learned a lot in a short amount of time. And then it was also just great to meet people. I got the chance to meet a few scientists that I had connected with over Zoom, but a lot more that I hadn't. So it was a pleasure to feel like part of the community for sure.
Definitely. And I know in the field of anti-doping, there are a lot of unanswered questions, and you're tapping into some really important ones with the work that you're doing, but there are often limitations, whether it's funding, staff, feasibility, time, you name it. So if we took away the barriers that are holding you back, Mike, what is the one problem in anti-doping that you would most want to solve?
Well, I don't know if this is unsolved just because of staffing or funding barriers or anything like that, but the scientific problem that motivates my whole lab is just that we cannot comprehensively measure small molecules in the world around us. We have this great analytical technique, mass spec, that allows us to detect small molecules with incredible sensitivity, which of course you're seeing in the doping field. That sensitivity is causing problems of its own, but for me as a computational scientist, the fact that there's so much rich data in a mass spectrometry experiment that we just can't interpret this incredible basic science challenge. It has huge ramifications throughout so many different fields of basic and applied science and medicine. So this is the problem that my whole lab is trying to really make a dent on in one way or another.
Definitely. Do you have thoughts if you had unlimited resources? What are some things that you could do to maybe make a bigger dent in this problem?
We're always constrained by the amount of data available. I don't think anyone working in a field of biology, chemistry, or medicine has a data set like the entire internet to train on, right? So I think if I had unlimited resources, putting that money into just data generation would be very helpful. That would solve at least some of the challenges on the analytical side. Now, of course, predicting what future chemists are going to think up and introduce into the gray market, that's maybe a bigger problem.
Well, Mike, we appreciate you sharing some of the work that you're doing and some of your insights with all of us today. But if you've piqued the interest of our listeners and they want to learn more about you and your work, where can they go for more information?
I do have social media. They could find me on Blue Sky and Twitter and my Google Scholar. And maybe by the time this podcast comes out, the lab will have a website, although that's sort of been sticking around my to-do list for quite a while now.
That's how it goes sometimes. That's all right. Well, listeners, definitely take some time to get connected to Mike on Blue Sky. Reach out to him if you have any particular questions. And Mike, thank you again so much for joining us on the show today.
It was a pleasure. Thank you for having me.
It was wonderful to chat with you. And listeners, great to have you here with us as well. If you haven't already, please take a moment to rate our podcast, leave a review, and subscribe on your favorite podcast platform. And please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.